Let's go ahead and get started. My name's Lynn Heasley, and I have the pleasure of introducing Bob Doebele for his talk today, as well as moderating any fights that break out afterwards. Hopefully there's none. Actually, this is Bob is coming up on 10 years being here. Actually, he came here in 2008, and he probably would have started the interview process in 2017, and I recall that, in fact, there was a high level of organization such that he came out on Martin Luther King Day, and Rafe Nemanoff was still over on the old campus, and they met out here but couldn't get into any buildings because, of course, it's on complete lockdown on a holiday here. So they finally managed to get into the hospital somewhere and have an interview. But obviously things went well enough that Bob agreed to come here, and it's been a delight to have him in our lung cancer group. Over these years, he's had a rather steep trajectory, and I would say at this point, he's at the tier of lung cancer investigators around the country that include Passiani and Trevor Bavona and John Haymack around the country that are probably poised to be the next movers and shakers in lung cancer in this next generation. And I've kind of come to that age in my career where I'm interested in what makes some people's careers work so well and why others get lost in the weeds, kind of like me. And if you kind of dwell on that, Bob came preloaded with a very solid project on low molecular weight G proteins. But in fact, some of us who were on his mentoring committee soon saw that it was like wading through mud uphill. Waist deep mud, and he was clever enough to, ALK fusions were just being observed in lung cancer at about that time, and he had the wits to start a project on that, and took the good advice to move into it completely, and he hasn't really looked back since as a molecular oriented physician scientist in lung cancer. I would say some of his strengths are he quickly identified the skills that are around him and moved his fusion kinase studies, both basic science forward as well as the translational and clinical components. And Leila Garcia with her excellent fish probes and the ability to make a fish probe within a couple of days on anything, and Dara Eisner and Ross Camage quickly formed a team, and I've had the benefit of sitting in on these occasional meetings, and it's crystallized team science. I've never seen it done better. If you didn't know, you wouldn't know who's the leader of the outfit, because in fact there really is no clear leader within this particular group. And they've really, over the years, we've now become a destination institution for treatment of fusion kinase and EGFR mutant lung cancer. So this didn't just happen because everybody's treating these. It's because we had the expertise and the basic science driving it. I would say another real strength of what's driven Bob's career upward so quickly is that he, in addition to a basic research lab that is very well fitted and suited, he uses his MD and his clinical attending duties to actually make his lab and the clinic seamless. And in fact, when he started out, there was 2 ALK mutant lines and 1 ROS1 line, and he's made it his business to have very interesting and important clinical IRB-approved protocols to capture biopsies from patients and establish cell lines and xenografts that have really allowed basic science inquiry to be connected with clinical treatment. So, this is the sort of stuff you're going to hear today. And Bob, I'm anxious to hear.
Well, Well, thank you for that very kind introduction, and I will definitely say that you asked the question of what makes a good trajectory, and having a fantastic team around you has really been the key to any success that I can claim. And looking back after being here for almost 10 years, I'd say that this is one of the best decisions I ever made to come here. The second best decision I ever made was listening to Lynn and Rafe when they told me to drop the low G GTPase project that I was working on. So thank you again for that salient advice. So today, I'm going to actually focus, there's a huge team effort. We'll acknowledge people along the way. A lot of what my lab has been doing over the last several years, trying to integrate this into a theme around why oncogene-targeted therapies work and why they fail, hence the title here. So this is the outline. Of course, if we're going to talk about drug resistance and why they don't work, first you need a foothold into why they work in the first place. So we'll start off with a story about developing a new oncogene-targeted therapy. We'll talk about kind of what most people think about when they think about resistance, acquired resistance. Many of you will be happy to know I'm going to actually have a short section that does not have to do with fusion kinases or gene fusions on intrinsic resistance. And then we're going to get into what I think is a somewhat newer idea, which is adaptive resistance, and I'll explain that in a second, and then some of the ways that we might approach this using both clinical strategies to understand adaptive resistance and also new models as well. So you've all seen this pie chart a million times, and it's grown remarkably over the years. I think I have to update this at least a couple times a year now. And I've highlighted some of the molecules that we're going to talk about today in terms of genetic subsets of lung adenocarcinoma. And I am going to bounce back and forth between multiple different models, ALK, ROS, RET, TRAC, and EGFR. And I think, having done this now for a long time, there are a lot of similarities in terms of how patients respond and how they become resistant to these therapies. So even though I'm going to bounce back and forth between the models, I think that is justified because the models have a lot of things in common, even though they're driven by different genes and we're treating with different drugs. And I'll point out that we still don't know all of the drivers in lung adenocarcinoma, but the piece of this pie is getting smaller and smaller. So I thought I'd try and define these different types of resistance right up front, because I think one of the things that I've struggled with in getting our papers reviewed and other things is describing actually what type of resistance we're talking about. So of course, if you're thinking about resistance, it helps to start with a tumor response. So if you have a patient with an oncogene target, they start off with a certain amount of tumor volume, you start treatment, they often feel better within a few days to a few weeks, and their scans get better. Their disease often stabilizes for quite some time, but ultimately, of course, and unfortunately, these patients start to progress again. And this is what we typically talk about when we are describing acquired resistance. And this is the point at which we often biopsy the patients to drive an understanding of why they became resistant, and probably represents some amount of clonal outgrowth of some mechanism of drug resistance. Intrinsic resistance is also relatively easy. So this is a situation, and again, there are different ways to define this. The example that I'm going to give today is an oncogene-addicted model that does not respond to our therapies because of a kind of a pharmacology issue. There are other types of intrinsic resistance, or patients who are intrinsically resistant to immunotherapy or chemotherapy, but here I'm talking about patients that we think should respond to these inhibitors, but don't. And then what I'd really like to kind of finish the talk with is this idea of adaptive resistance or residual disease, which hasn't gotten a lot of attention. And you'll see perhaps from some of the graphs that I show you later in the talk why so. I think we've been so enamored with the responses that we haven't thought about why we don't kill more of the cells in the first place. And a lot of this came through my discussions with Paul Bunn over the years about log cell kill and why we aren't doing a better job of this. And so this is, I've intentionally not drawn this stable disease here down to zero because it's very, very rare that we eliminate all the tumors, even on the scan. And even when we do eliminate all of the radiologic disease, we don't always, it typically comes back. And of course, there's a lot of variation in this from patient to patient, meaning that how low we go at maximal response is different. And again, we'll kind of show you some data support this idea later on in the talk. But this is what I'm going to talk about today. So response, acquired resistance, intrinsic resistance, and adaptive resistance, and give you examples that we've developed models from our lab that have kind of illustrated some of these points. So again, it helps to start with what is actually a good oncogene-addicted model and a response to therapy. And again, I'll try to highlight people in my lab that have helped with this. There are many, many others behind the scenes, but I really like to highlight their work today, and this is done mostly by Aria, who recently defended her thesis and now is off at the Huntsman Cancer Institute with Martin McMahon in a postdoc position, and An Lee, who's still stuck with me. So as I mentioned previously, there's a small part of that lung adenocarcinoma pie that we still don't know who's driving those genes, and we thought that would be a good place to look for new oncogenic drivers. This is a bit of an old story, so I'll kind of go through this quickly. And we identified two putative new oncogenes that were variations on each other, fusions involving the NTRK1 or the nerve growth factor receptor version A. These look like putative gene fusions by the in-frame sequencing that we saw, but of course we wanted to prove that these were indeed oncogenic, so we cloned these genes, expressed them in model cell lines, and showed that they were phosphorylated, autophosphorylated, that they could drive MAP kinase signaling. Furthermore, that they could drive cell proliferation in a BAF3 model when you withdraw IL-3, and most importantly, that they could form tumors in mice. And of course, we didn't want to just identify new oncogenic drivers. We want to be able to drug these, and it helps to have better models than BAF3 expressing an oncogene model. Those will suffice for some purposes. So we were fortunate enough from this one of the first patients we identified here to derive a cell line from the patient's pleural effusion. So not only did we derive a cell line, we derived a patient-derived xenograft that went straight into mice with the help of Antonio Jimeno. Turns out that the PDXs grow very slow, so we haven't used them for much in terms of drug treatment, but the cell line was very robust and amenable to study. We also identified two other existing cell lines, a colorectal cancer cell line from the NCI60 that has another version of an NTRK1 fusion, and an acute myeloid leukemia that has an NTRK3 fusion. And NTRK3 is a very related receptor tyrosine kinase with near identical sequence at the ATP binding site. So any drug that should work for one should work for three, and indeed actually work for NTRK2 as well, though those are rare fusions in cancer. So to demonstrate that we could target this, we did simple signaling studies with a drug initially called Array 470, now known as Loxo 101, as it's spun out into a company in clinical trials. But you can see very nice target inhibition of the TRK kinase and the fusion kinase for all three of these cell lines at low nanomolar doses, ranging from 5 to 50 nanomolar, and concomitant downstream signaling inhibition of ERK and AKT in most of these cell lines as well. And as we would expect, these cell lines, each of the three different cell lines harboring different fusions and different histologic subtypes, all respond to this therapy. And again, getting back to the point that the IC50 should be nearly identical given the structure of the ATP binding site, these BAF3 cells expressing either an NTRK1 or an NTRK2 fusion have nearly identical IC50s to the same Loxo 101 compound. So we are very encouraged by this. It's always nice to have xenograft models, and fortunately all of these cell lines implant into mice. And we saw, again, very good results. So untreated controls grow very rapidly for all three xenograft models. And at either 60 mg per kg or 200 mg per kg, we see either tumor stasis or significant regression in 2 out of the 3 models. And so again, this was very encouraging and gets to the idea that, you know, perhaps the origin of the tumor doesn't matter. Not only are there 3 different oncogenes here, these are all derived from different types of tumors, lung, colon, and leukemia. In a lot of ways couldn't be much more different. And indeed, these TRAC fusions are not particularly common in any disease. Probably the most common disease are these rare tumors, childhood tumors or mammary analog secretory tumors where they may define the tumor, but for the most part, they're pretty rare subsets of lung sarcoma and other tumors. And this has led to an idea that I am hoping will take hold, although that has not been evidenced yet, at both companies that are promoting their TRAC inhibitors are going to the FDA with a molecular label. So the idea being that it's not going to be TRAC fusions in lung cancer or TRAC fusions in colorectal cancer, it's just going to be TRAC fusions. And that, to my knowledge, has never been done before. So this may be a first in terms of targeted therapy, not having a label that's specific to a certain type of cancer, but just to the genetic alteration. And of course, I was unbelievably fortunate to be able to test this with my own patients. So the very first patient we identified with a TRAC fusion happened to have sarcoma, and she lived in Portland, but we were the closest site for this brand new clinical trial, and so she came here quite ill, actually, when she started. As you can see from her scans, her lungs were filled with in the range of 40 to 50 tumors, but she's seen a very nice response at 2 months and then ongoing, and she sends me updates every once in a while, and she just actually passed her 2-year mark last month, still on the same therapy, no evidence of resistance. Does this only work for her? No. The trials are not releasing their phase 2 data yet, but they have released their phase 1 data. These are just looking at TRAC-positive patients. Again, a number of different tumor types, as you can see here: colorectal carcinoma, lung cancer, mammary analog secretory carcinoma, brain tumors, and the responses are quite good: 83% for LOXO-101 and 100% for the 3 evaluable tumors in the atrectinib trial. These are, again, from the phase 1 studies. The phase 2s are now accruing worldwide and ongoing, hopefully with presentation to the FDA as early as 2018, which is not bad for an oncogene that we kind of discovered and developed or discovered in lung cancer and developed a therapy for starting in June of 2012. So we're quite excited about this. And again, highlights when you have the right marker and an appropriate selective drug, you can get really remarkable targeting results for these patients. So that's the good news. Unfortunately, there is a lot of resistance in cancer. And so we'll move on next to the idea of intrinsic resistance to oncogene-targeted therapy. And again, this is some of you may not know these people yet, but Adriana Estrada Bernal is a research associate in my lab who's done most of the work in these models, derived all of these cell line models. He's really quite an artist at nurturing these cells until they develop into cell lines. And Andy Doak is a new PRA in my lab who came to us from CU Boulder and is helping out with this project as well. So there are some oncogenes that we can detect, but we can't necessarily treat very well, and those include EGFR and HER2 exon 20 mutations. So, of course, you all know that we have good therapies, erlotinib, gefitinib, and afatinib, for patients who have the common EGFR mutations, exon 19 and exon 21. There's, again, 3 FDA-approved drugs for those. When they develop resistance, most of them do so through a second site mutation at T790M. AstraZeneca has developed a nice compound for that, osimertinib. There are others in the works with excellent response rates. So for those patients who have one of these classical sensitizing mutations and who develop resistance through T790M, we've got great treatment options. Unfortunately, there are a small subset, about 10% of EGFR mutations that occur in exon 20, and then a smaller set or a similar set in exon 18 that have variable responses to EGFR TKIs, and none of them have FDA-approved therapies. In fact, sometimes the label restricts this, and the insurance company tells us we can't use these drugs. I'm not going to talk about HER2 mutations, but they suffer the same fate as these patients. To my somewhat surprise, despite the fact that there are literally hundreds of lung cancer cell lines, not a single one in the banks made by Adi Gazdar or John Minna or others actually had an EGFR exon 20, making it a bit difficult to study. So all the models were in BAF cells and other cells. And as you can see, there's quite a range of different exon 20 insertions. So the most common are highlighted here in red, insertion of ASV, an insertion of NPH, an insertion of SVD. These are all very common in terms of EGFR exon 20. It turns out these are actually sensitive to standard EGFR inhibitors, the insertion FQEA. Luck would have it that the first 3 cell lines that we developed match the 3 most common EGFR exon 20s. And again, on lead to that work, and my clinical colleagues helped me identify these patients for cell line derivation. So here's both the preclinical and clinical evidence that our current EGFR inhibitors really don't work. So this is in vitro expression of these molecules, which is what we had to do until we had our cell lines, demonstrating that the EGFR exon 20 insertions respond quite poorly to standard EGFR inhibitors. And this is the clinical evidence showing progression-free survival with a Kaplan-Meier curve that looks like you're kind of tumbling down a cliff here with very rapid progression on these drugs because they just don't really hit the target. And the problem has to do with the position of the mutation. So these insertions in the C alpha helix of EGFR make it resemble wild-type EGFR. And so you can inhibit these molecules just like you can inhibit wild-type EGFR with drugs. The problem is it takes doses that are too toxic for our patients. So Ariad, who we've actually worked with to develop one of their compounds, and they've used one of the 3 cell lines that we have, took one strategy, I'll show you another in a second, which is to try and thread the needle between these EGFR exon 20 insertions and wild-type EGFR. So what they've shown here are IC50s relative to wild-type EGFR, so that's in the green for each drug, erlotinib, gefitinib, afatinib, and their new compound, and then showing the IC50s against a variety of different mutations, including the EGFR exon 20 mutations, which are clustered towards the right side here. And you can see that erlotinib and gefitinib can overcome these, so can afatinib, but they do so at doses well above wild-type EGFR, and that's limited by our patients' toxicity. So we just can't give doses that are that high. They've designed a drug, they think, that they can thread the needle, but you can see that there's very little clear white space between this dotted line and those IC50 values, suggesting that they've got a tough task ahead. Still in phase 1, so we don't know, but this will be a challenging needle to thread, as I said. This is data that they derived with our cell lines. This is the CUDO14 cell line, demonstrating that AP32788, their phase 1 compound, inhibits phospho-EGFR and downstream signaling through MAP kinase in the range of about 100 nanomolar, whereas osimertinib, a third-generation EGFR inhibitor that AstraZeneca is also looking at in this complex, really you need to go out to at least 1,000 nanomolar, probably doses that we can't achieve in humans. And when you benchmark all of these drugs against each other, Ariad's AP32788 looks a lot better in our cell line with an IC50 that's low, but again, still a very narrow window. And the question is whether they can achieve doses that are far above this without causing toxicity to our patients. And as I said, that trial is ongoing, and we're one of the lead sites for that phase 1 trial. What about other tactics? And so, we obviously these patients are desperate for a therapy. And in collaborating on a clinical trial some time ago, we stumbled upon a compound that might have a kind of novel way of inhibiting this. So this is a PET CT with a marker that tracks hypoxia, demonstrating that at least in this particular patient's lung cancer, that there's high levels of hypoxia. And of course, that's associated with a lot of tumor types and can drive proliferation, metastasis, and angiogenesis, and a lot of features. So this molecule, as you'll see, is going to be somewhat dependent on a hypoxic tumor environment. So these clever collaborators at the University of Auckland, this is Jeff Schmale designed the drug on this on their platform, and Adam Patterson did a lot of the early preclinical work. And then we've kind of adopted this as testing this in our models to try and overcome the inherent EGFR resistance to these therapies. So it has a reactive side group here. So this is tarlaxetinib bromide. TH4000 is the prodrug. TH4000E is the active drug. Under normoxic conditions, this side chain is stable and prevents binding to any EGFR molecules, or at least significantly reduces it. In the presence of hypoxia, this undergoes fragmentation, and the idea being that this is a way to get very high levels of this drug in the tumor, but not in normal tissues. This then becomes a covalent EGFR inhibitor, actually very similarly related to afatinib, one of the clinical compounds. But again, the idea being here that the dose levels in the tumor are going to be far, far higher than they are in the normal surrounding tissues. Again, this covalently attaches and inactivates EGFR. And so we had this idea, well, this would be a great model to test this because, there are lots of EGFR inhibitors, but this would be, EGFR exon 20 insertions would be a great place to test this because all we need is higher doses. We don't necessarily need something special to inhibit these molecules. And so this is comparing afatinib, TH4000E, these are under normoxic conditions, and gefitinib, so this is the active metabolite, so the cleaved drug. And you can see, actually, it doesn't look all that special. You see inhibition of phospho-EGFR and downstream MAP kinase and AKT at a very similar dose. And in fact, in this experiment, it's a little bit worse than afatinib. So you can see that there's only partial inhibition at 10 nanomolar, but more complete inhibition of afatinib at 10 nanomolar. Gefitinib, of course, you have to go out to about 10 micromolar before you get significant inhibition, or at least 1 micromolar. So in terms of the active metabolite, it's not actually that special of a drug. And again, that's highlighted here again when you look at proliferation assays. So now looking at all 3 cell lines, you can see the red and the blue lines. So red is afatinib, blue is TH4000E, that's the active metabolite. They track very similarly with similar IC50 values. They're far better than gefitinib, one of our clinical compounds. And far better than the prodrug, because this is under normoxic conditions. So you can see that the prodrug has IC50s again in the 2 to greater than 10 micromolar range, again highlighting the mechanism of action of this drug. It just doesn't inhibit EGFR very well unless it's cleaved to its active metabolite. And of course, the real proof is going to be if we can get this into patients. But the next best thing is to try this in xenograft models. And we were quite pleased with our early data in 2 of the 3 xenografts. The third one we're having a bit of trouble getting to grow in mice. But in these 2 models, 2 different EGFR exon 20 insertions, I'll highlight the one on the left first. You can see that afatinib, given at doses that mimic the PK and exposure of humans, really has 0 effect. And that's pretty consistent with my clinical experience of using this drug in patients, that it looks similar to placebo or control. Cetuximab actually had some modest effect. So this is a monoclonal antibody that binds to the extracellular domain, doesn't care what kind of mutation there is. And we've actually seen some anecdotal evidence, although there's never been a good clinical trial of this drug in EGFR exon 20 patients. This might be something that we follow up on with a clinical trial. And notably, afatinib plus cetuximab here in yellow looks just like cetuximab. So afatinib's really not doing much. But TH4000, and again, I'll highlight that here we're giving the prodrug, right? So this is going to be dependent on cleavage to TH4000E, actually causes tumor regression. And here, we didn't have enough mice to do the full experiment, but you can see that the TH4000, again, at least causes tumor stabilization, not necessarily regression compared to control. So we're very encouraged by these results and actually working to get this drug back into clinical trials for this specific indication. One of the things that we've really tried, because again, we're the only holders of these particular models, and that's true of other models as well, is getting them into the hands of other collaborators. So again, sometimes we do the work in our lab, sometimes we ship it out. So we've worked with New Zealand on their compound. We've worked with a different small molecule inhibitor at MD Anderson called poziotinib. We've given these to investigators at UC Davis to look at combination therapies. Broad, Ariad, I mentioned. Paul Huang is using these to look look at proteomic profiling of these cancer cells to see if there are other liabilities. And then we're in talks with the Cancer Institute in New Jersey and University of Cologne to get these in their hands as well for ideas that they have on treating these drugs. And we've been, we've tried to be generous with these models for other of our cell lines as well, where we have a unique molecular alteration. Acquired resistance to oncogene-targeted therapy is, again, something that you've heard me talk about. And this, I think, highlights some of the kind of themes that Lynn was talking about, the bench-to-bedside or bedside-to-bench research that we do. And again, many more people involved in this, but I thought I'd talk about work specifically Sarah Nelson, who just defended her thesis in my lab. It has actually just started back in the clinics yesterday. Caroline McCoach, who's a clinical fellow in my lab, who's helping with gathering resistance data, and who actually does a lot of the sequencing of our resistance samples. And again, this is really kind of a circular project. And I'll give you examples of both where we go from, we start with a lab-based model and identify putative mechanisms of resistance, or we identify something in the clinic that we're not sure whether it's resistance and go backwards. And this is work that Caroline and have been collecting. These are a series over the last 5 or 6 years of ALK and ROS1 resistant patients. Here is highlighted the ALK resistance data. When we first started, it was only resistance to crizotinib. And this is, I think, a little bit sobering, is that, as opposed to EGFR resistance, where you see the majority getting a single site mutation, for crizotinib, only a small proportion, about 25%, were getting resistance mutations, and they were kind of scattered all over the molecule. Since that time, a number of second-generation inhibitors have been developed, and you can see the resistance mechanisms after more than one TKI highlighted on the right side. You can see that now the resistance mutations are greater. I think there's actually some bias in our data here. We're trying to work to see whether we can explain this. There are a lot of reasons why the profiles of resistance might change, but one reason is that if you take a patient who's resistant to crizotinib and they don't have a resistance mutation, they don't respond to therapy, we're very unlikely to rebiopsy them again 6 weeks later after they fail another therapy, because the assumption is that they just have intrinsic resistance, and therefore the denominator is different in this set. But either way, there are a lot of mechanisms of resistance that occur either outside of the primary target or just completely unknown. And this is, I think, going to be a challenging problem as we kind of treat our patients with more than one sequential TKI. And a lot of these patients have received 3 or 4 ALK TKIs. We're also seeing increasing complexity in the mutation. So when we first started this work, we'd only see a single mutation. Here we see an example of 2 mutations arising in the same tumor sample. They occur at a nearly identical varying allele frequency. And in this particular case, we know they're in cis because they're close enough on the sequencing reads that we can identify both mutations across single reads. So this is a complex mutation. This molecule, this particular ALK molecule, should not be resistant to the drug this patient was on if they had either one of the mutations, but the double mutations have never been tested, so that's something new that we have to start looking at, is complex mutations. We are also beginning to see, perhaps as we get more sensitive techniques, evidence for different clonal populations. So this was a ROS1 patient, also with 2 different resistance mutations, but here, at least suggestive of 2 different populations. There's 2 different varying allele frequencies, and we had at least, the cell line unfortunately never grew out, but we had an early cell culture where we were able to treat with crizotinib, which shouldn't hit either of these, and it doesn't, so there's no decrease in phospho-ERK, or ceritinib, which should hit the gatekeeper mutation at L2026M, but not the L1951R. That's from prior work that was published just about a year ago. And here we see only partial sensitivity, and we think this was due to a mixed population of cells. And ultimately, at that time, there was only ceritinib to put this patient on, and they didn't respond and then had a fairly rapid progression, which would be consistent, although does not prove that there was outgrowth of this 11-1951 clone, which does not respond to ceritinib. We're also looking at new ways to analyze the data. This is still somewhat preliminary, but now that we're doing a next generation sequencing panel, we can try to gather data on how many unique reads there are, which may be evidence of copy number gain. I'm only showing part of the gene panel here just to highlight a few cases. These are blue cases or ALK resistance cases, red or ROS1 resistant cases, and the ones that are gray are other samples that don't fit into that category. You can see for the most part that these tumor samples cluster very nicely for each gene, and the black line here represents the 3 standard deviations above the median for these samples, and you can see that there are several outliers, and we're trying to identify whether these may correlate with resistance mechanisms. It's noteworthy that this ALK patient also has a concomitant KRAS mutation and also seems to have high copy number of KRAS. This patient does not have an ALK kinase domain mutation, but has evidence of increased copy number of a GTPase subunit that may be increasing MAP kinase signaling, and others as well. But we're still working to validate some of these results. One of the things that we've identified, again, thinking about resistance mechanisms that we identify in a patient, but it's hard to prove whether it's true resistance or just some bypass mutation, is this example. This was a case that was sent to us actually from a colleague in Poland, Rafał Żyżewski. You can see here, this is a ROS1 positive patient with a large tumor in the right lung, nice response to crizotinib, but ultimately progression here that was biopsied, was sent to us for sequencing. We found a KIT mutation, but again, we don't know if that was resistance or just some bystander mutation. So we cloned it and demonstrated that first of all, this was an activating mutation in KIT, so it's autophosphorylated, and that you could at least drug it with reasonable, well, not reasonable, but high-ish doses of a drug called ponatinib, and has similar activation to a previously identified substitution at the same point, D816V. But to show that this could actually induce resistance to crizotinib, we cloned this into one of our cell line models that we derived here, CUDO2, which is a ROS1 model. So you can see that the empty vector has a reasonable response to crizotinib, or dose response curve, but once you put in this activating mutation in KIT, they're completely resistant to crizotinib, consistent with what we saw in the patient. And that more importantly, we could dose this now. If we add in 500 nanomolar ponatinib, we can resensitize these cells to crizotinib, suggesting that this is a mechanism of resistance, and now we know to look for this in ROS1 and other patients as well. So not all panels have these alterations, but this is now kind of added to the catalog of many different resistance mutations. So that was an example of where we started with a clinical sample and kind of went back into the lab and modeled it. This is where we're going the other direction. So unfortunately, we still don't have great clinical RET inhibitors. I just started my first patient this morning on a novel RET inhibitor, so I'm hoping we finally found one. But until that time, we were still interested in understanding how our models may become resistant to RET inhibitors. And so we have only one cell line. I'll come back to this later in the talk about new models that we're deriving. So we only have one cell line, an LC2AD cell line that has a particular RET fusion. You can see that that cell line and a thyroid cancer cell line that has a RET fusion, TPC1, are exquisitely sensitive to ponatinib in the range of about 10 or so nanomolar, and far more than a cell line that does not harbor a RET fusion. And as you would expect, these RET inhibitors decrease phospho-RET as well as downstream signaling. So we took two different paths. We just wanted to try different methods. Nothing special about this. We thought maybe this one might be, this method of kind of a steep increase in dose right away was more indicative of how we actually treat our patients. We don't slowly step up their dose, although most of the models, most of the labs that do resistance slowly step up the dose of drugs, starting with very, very low non-inhibitory doses. So we did both, and we got two different clones out, ponatinib resistant 1 and ponatinib resistant 2. Again, both derivatives of the LC2AD RET fusion cell line. You can see that they're both markedly resistant to ponatinib. compared to the parental cell line, so PR1s in red and PR2s in green, and similarly resistant. We tried other RET inhibitors as well and demonstrated that they were resistant to cabozantinib and other RET inhibitors, so that's consistent. And unlike the LC2s, where you see nice decrease in phosphorylation of RET and downstream signaling, you see no decrease in downstream signaling of phospho-Schick, phospho-ERK, or phospho-AKT. What's also a little bit interesting and surprising, I didn't put the figure in here, but you'll notice that at baseline, before we treat with drug, there's actually no phospho-RET, suggesting that something is actively downregulated. So the protein is still there, as you can see. There are no kinase domain mutations in these, of course, in RET. And data that I don't have time to show, if we treat with a phosphatase inhibitor, this comes back on, suggesting that there may be a phosphatase that's actively turning off RET so that someone else can signal. So one of the clones was relatively easy to identify, and we actually identified it through next generation sequencing, but what I've shown you here is the direct sequencing. So there's a point mutation that causes a known NRAS Q61K mutation in PR1, but not PR2. We actually found both through RNA-seq and other targeted sequencing methods, we weren't able to identify any activating mutations in a known oncogene that would account for the resistance in PR2, again, including resistance mutations in the RET kinase domain. We actually had, so the first thing we always like to do is kind of re-express the mutant form back into the parental to show that it becomes resistant. We actually couldn't do that in the LC2ADs, but we could do it in the TPC1 thyroid cell line. And you can see using a RAS pull down here that introduction of the NRAS Q61K mutation increases GTP loading for NRAS. It seems like these cells might be dependent on a different isoform, although we haven't chased this up yet because the active GTP-bound RAS here is a lower molecular weight than what's seen in these others. And then again, going back to that idea of the phosphatase actively deregulating RET, we needed to transform or put these mutant NRAS in the presence of a low dose of ponatinib to suppress RET signaling. They wouldn't switch over to NRAS unless we turned off RET. And may have something to do with why we couldn't get that into LC2ADs. And again, in this cell line, the TPC1s were markedly resistant to ponatinib when they were expressing this activated RAS. And as you might expect, so RAS can regulate MEK. We wanted to know whether knockdown of RAS was important. So in the parental cells, surprisingly, MEK NRAS knockdown was not important, again, consistent with that idea that they may be dependent on a different RAS isoform, but they are sensitive to RET knockdown, whereas in the PR1 cells, they no longer care about RET knockdown, but they do die when you knock down NRAS. And consistent with that idea, the PR1 cells are more sensitive to a MEK inhibitor, in this case trametinib, than the parental cell lines are, again suggesting that they've kind of switched their dependence from RET to NRAS, and that the resistance is mediated mostly through MAP kinase signaling. The PR2s were a little bit trickier. I won't go into all the background on how we ultimately figured this out, but they switched dependence to EGFR. You can see that the PR2s have markedly increased EGFR expression as well as phospho-EGFR. In proliferation assays, they are only minimally sensitive to EGFR inhibition, but you can see that they are more markedly sensitive than the parental cell lines. And I think what's more convincing here is the signaling data on the right. So when you treat with an EGFR inhibitor in the parental cells, there's really not much of an effect on downstream signaling, but in the PR2s, you see marked decrease in phospho-ERK and phospho-AKT. And again, though, this was only partial. Again, this is mostly from our RNA-seq data, and this is actually work that Ike Chun did, helping us analyze the RNA-seq data in his lab. We identified a second potential mediator of resistance, which was Gas6 and AXL. Unfortunately, we still don't have very selective AXL inhibitors, but you can see that you can further reduce the sensitivity to afatinib. So this line is just afatinib dose response alone. If you use two different arguably multi-kinase inhibitors, cabozantinib or furetinib, you markedly reduce sensitivity, and we believe that this is through AXL. Kurt Davies in my lab many years ago had derived a resistance model that was very similar for ROS1, the HCC78TA684 resistant cell line, that also had this kind of modest sensitivity to EGFR. And again, when we went back and retested those with the addition of an AXL inhibitor, again with the caveat that these drugs hit multiple targets, it markedly enhanced sensitivity to them. And so this to us suggests that AXL and EGFR are cooperating to derive resistance in these cell lines. And this, I hope, will be a nice segue into the kind of adaptive resistance. So going back to the parental cell lines, we were curious about the expression of EGFR and AXL. It turns out the ROS1 cell lines, which I haven't shown here, have a bit at baseline. The RET cells don't have a ton of expression at baseline. You can see before we start to treat with a very low dose of ponatinib. But look what happens to EGFR and AXL expression within 48 hours of inhibiting RET. You get massive upregulation of both EGFR and AXL, suggesting that is a very early and adaptive response. Again, these are not the resistant cells. These are not the fully acquired. These are the parental cells just treated with very short term. And so this is, I think, a good lead-in into this idea of, there are probably early concomitant signaling pathways that we may be able to target that will reduce the ability of these cancer cells to survive and adapt to our targeted therapies. So moving on to the next section, almost all this work was done by Aria, who, as I said, is now off in her postdoc at the Huntsman, but is now being picked up by Andy in our lab, who's going to carry on the torch and help us understand this adaptive signaling better. So again, I won't go into all the reasons that we focused on EGFR, but I think this is one good reason. So this is a 1993 publication from Valerie Rush, who's a surgeon at Memorial Sloan Kettering, looking at EGFR and EGFR ligand expression in non-small cell lung cancer, showing that the vast majority express this. It's probably one of the seminal articles that made OSI and others develop EGFR inhibitors before we knew that EGFR mutants existed, or EGFR mutations. And similarly, when we look at ALK and ROS positive patients, we see that they also have very high EGFR IHC expression. So we collected this data at a time when we were still doing EGFR IHC in the clinical lab. You can see that most samples actually have the maximum H score of 300, and most of them are very high with very few low numbers. And they're very similar to patients who have KRAS mutant or non-mutated lung cancer. They have very high wild-type EGFR expression. That thought made us think that there must be something there. And so we wanted to know, again, whether in naive cells, not acquired resistance, but naive cells, whether the addition of either an EGFR ligand or an EGFR inhibitor could modulate the response to a fusion kinase inhibitor. So this is a ROS1 cell. The FDA-approved drug is crizotinib. If you take these cells and just treat it with the FKI alone, crizotinib, you see this nice dose response. If you add an EGFR ligand, such as EGF, you get resistance. But notably, if you add gefitinib to that, or in the FKI alone, you see resensitization. And that doesn't just occur in one cell line. We've expanded this to multiple different fusions, so several ALK cell lines, RET cell lines, and TRAC cell lines as well. And they all show a similar pattern, although to varying degrees. When we dig into the signaling reasons for this, we see that combination therapy, meaning the specific fusion kinase inhibitor, and in this case gefitinib, we see enhanced MAP kinase signaling, so either looking at phospho-MEK or phospho-ERK, and also typically phospho-AKT, suggesting that wild-type EGFR, even though it's not the primary driver, is contributing some kind of minimal signaling, and when you inhibit that, you markedly reduce that signaling. Now, the mechanism for this, the easiest mechanism to explain is that EGFR is just signaling on its own. There's either ligand in the media or in the case of an actually true human tumor, it's coming from the microenvironment, and it may just be true bypass signaling. But we also uncovered some novel mechanisms that we think may be contributing to the role of EGFR, and this was a bit more surprising to us. And this is an example of 4 different fusion kinases, TRAC, ROS, RET, and ALK. Even in the presence of a dose that would normally inhibit the fusion kinase, as you can see here, loss of phosphorylation, when you add EGF in, you can actually rephosphorylate the fusion. So it's not just signaling around the fusion, it's actually somehow interacting with it to rephosphorylate it and activate it. So that's one mechanism, perhaps direct interaction, and we've shown that they co-immunoprecipitate EGFR in each of these fusion kinases, both in cell lines natively expressing both of these molecules or by co-expression studies. The other mechanism was also kind of interesting, and that is what we have fondly called adapter switching. And so for this, we've used a proximity ligation assay to measure complexes between a kinase and adapter. They can really be used for any 2 proteins. And basically what we're showing here is that at the beginning of the experiment, the 0 time point, you see evidence of ROS1 GRAB2 complexes. So every red dot in theory is a complex or cluster of complexes between the ROS1 protein and its SH2 adapter GRAB2. And then we treat it at very short time intervals, 5, 15, 30 minutes, and we see those go away as you would expect. So you dephosphorylate ROS1 and GRAB2 can no longer bind. What was less expected was the concomitant increase in EGFR GRAB2 signaling that occurred during that very short time window, suggesting that it wasn't kind of transcriptionally mediated or mediated by other methods. And you can see the quantification here. ROS1 GRAB2 complexes decreasing in blue and EGFR GRAB2 complexes increasing in red over a very short period of time, suggesting if you shut off one target, another can bind that adapter very rapidly. And this is reversible. So if you wash out the drug, you see EGFR over a couple of hours lose GRAB2 binding and ROS1 GRAB2 binding increases. And then as kind of our final experiment, we wanted to see whether this would work in terms of combination therapy in mice, and now we're actually trying to get this as a clinical trial. You can see that gefitinib has really little effect compared to control, as we would expect. It's not the dominant oncogene. Crizotinib stabilizes these tumors nicely in this model, and the combination of both an ALK inhibitor and an EGFR inhibitor cause a greater decrease in tumor growth combined together. Again, for the reasons that I mentioned earlier, gefitinib is a very poor wild-type EGFR inhibitor. And so we're looking at whether cetuximab, which may be a better wild-type inhibitor, or even drugs like TH4000 tarlaxotinib, may be a way to actually inhibit wild-type EGFR in combination with these fusion kinase inhibitors in patients who have not yet gotten acquired drug resistance. And then we've also shown that this happens in patients. So this is a cell line, again, a parental model of a ROS1 cell line treated to resistance. And you can see that this is the EGFR-mediated resistance. And you see these PLA complexes. And this is actually a patient before crizotinib therapy and post-crizotinib therapy. And we've done this with a number of ALK and RET patients as well, demonstrating that EGFR is present and can drive resistance. And so the idea, again, being here that crizotinib may shut off the primary target, but through a number of different mechanisms that I've described, EGFR can get around that and may lead to an incomplete tumor response. Of course, it doesn't cause, we think, complete resistance, but our idea is that if we then combined an EGFR inhibitor with ROS1 or ALK inhibitor, that we would get an enhanced tumor response, and that may delay progression. So how do we actually kind of look at this clinically? Well, you may not believe me that causing kind of greater tumor response would actually lead to better progression, right? A lot of these patients have great responses already. Is shrinking the tumor more actually going to lead to a better response? Well, I'll describe first Caroline's work that we've collaborated with the FDA to demonstrate this, at least in principle, and then some of the models that we're developing to help us answer some of these more difficult questions that Laura Schubert and An Lee in the lab have done. So the question that we started out with is a deeper response actually better? We don't measure that, right? We just measure whether they have a partial response. That's minus 30%. So it's this, I would argue, somewhat arbitrary line drawn right here. So we asked the question a different way. If you get a deeper response here, is it better than a response here or here or here? And so we collaborated with the FDA. They were able to get data from 2 clinical trials that use crizotinib as their treatment arm or study arm. Contained 305 ALK-positive patients, and we pooled the individual data, and then we just, and again, arguably arbitrarily grouped them into quartiles of no response at all, minor response, 2, 3, 4, getting deeper and deeper responses. And sure enough, when you looked at either progression-free survival or overall survival, the patients with the deepest responses did better. So again, the deepest responses going to no response at all in purple, and same thing for overall response. And I just put this in here because I think 3D graphs are cool. This is analyzing the data a little bit different way, but I think it does kind of drive home the point. So again, this is quartile 4, 3, 2, 1. This is average depth of response in those quartiles, and that's of course mandated by the way that we analyze the data, but this is duration of treatment. And you can see that a deeper response is better, and this is actually that same data at an individual patient level data. As you get deeper, on average, you get better and better responses. Not uniform for every patient, but in general. And so, again, kind of looking at our model, this is how we typically treat patients. We start with a tumor, we get an incomplete response, and then we biopsy at acquired resistance. And I think we're getting somewhat expected answers, but not all of the answers. So there's where we typically biopsy. The question is, how do we actually get to understand how we reduce this residual tumor volume here so that we can come up with more rational targeted therapies? Well, I already described one that we kind of came up with based on data from our lab, but it'd be really nice to really understand why tumors don't shrink. And for that, Caroline McCoach, who's a clinical fellow that's been working with me, has designed several clinical trials, one of which is open, and they're across different settings with different oncogene targets. So the first one that's open is an EGFR early rebiopsy trial. So in this particular trial, patients with late stage EGFR mutant lung cancer get a pre-therapy biopsy, so immediately before they start their EGFR TKI, and then 2 weeks of EGFR TKI therapy, and then another research biopsy where we collect frozen tissue. That one we've already started accruing to. The other idea is to move these therapies into early stage disease, because if we give them neoadjuvant or induction therapy, in this case crizotinib for ALK-ROS or MET, because that drug inhibits all 3 of those oncogenic forms, we can then resect the patient at 6 weeks and get a larger sample of tumor to study why they don't completely go away. And of course, this has been our ongoing procedure, which we've enrolled about 200 patients on our post-progression biopsy. I don't have tissue from all 200 of those, but that's been a very successful rebiopsy. And this is just showing data from 2 of the patients where we've gotten both the pre-treatment and the post-treatment biopsy. You can see that there's nice tumor shrinkage for both. We intentionally set the window somewhat small here because we're often starting with lesions that aren't particularly big, but you can see a very nice 2-week response to therapy. And we're in the actual process of analyzing the RNA-seq on these 2 patients right now in collaboration with Lynn Heasley and AC Tan and others. And finally, we're trying to generate new models. So, Sarah Nelson in my lab really lamented that we only had 1 RET fusion cell line. Of course, as soon as she left the lab and went off into the clinic, An was able to get another one going. So this is one way that we're trying to enhance our models and try to make sure that what we see in one cell line is representative of other cell lines. This is a brand new cell line that has KF5B RET. It's very sensitive to 2 RET inhibitors. But in thinking about some of the questions that we want to ask about residual disease and lack of response to immunotherapy, which we haven't talked about here, we were trying to develop mouse models that may allow us to kind of understand the accumulation of genetic lesions in early tumors, things that enhance tumorigenesis, metastasis, or inflammation. And to do that, we need immune-competent models. And so An and Laura in the lab have designed this system based on some others, but I think with some enhancements, notably Andrea Ventura at Memorial Sloan Kettering, who gave us some of the early EML4 ALK. Our goal is to make multiple different fusion models to correlate to some of the human cell line models that we have. And to do this, it's a little bit tricky. Laura's had to learn a lot about mouse genetics and the fact that the chromosomes, although there's high homology at the gene level, the chromosomes don't always line up well. So for example, one of the common partners of RET is CCDC6, and we thought that based on the chromosomal location, it just wasn't going to combine well with RET. But she designed small guide RNAs to cut KFIB and TRIM as well, KFIB and TRIM24, as well as RET. Miraculously, when you cut these with Cas9 using a guide RNA to intronic sequences, at some rate, they recombine into a fusion gene. And so we made multiple different small guides corresponding to human oncogenes in ROS, RET, and NTRK to make models to complement our ALK ones. And I think they came up with a fantastic strategy. Part of the hard part is before you invest in making adenovirus to inject down the lungs of mice, is knowing whether your CRISPRs are actually work. And so nicely, the BAF3s provide this great model. So first of all, they made DS-RED positive BAF3 cells using this lentiviral construct. And when CRE is expressed, although that's not shown here, they'll turn green. Because this is floxed. So first we started with the DS-RED selected cells, so we now have a pure population of BAF3 cells. And again, once we put small guides in with these CRISPR Cas9 constructs, they also express CRE, and we'll switch these to green. So to do this, we can make individual guide RNAs and mix and match them into the BAF3 cells. So this is the painful part of making these models. And we can assess whether our transfection worked by just determining what percentage of the cells become green, and so we can make sure that our transfections are working. But the real beauty of the BAF3 system is that BAF3 cells are typically dependent on IL-3, and so normally if you're doing this, you'd have to kind of screen either single cell clone or sequence large numbers of cells, use deep sequencing techniques to know whether your CRISPR guides actually generated the fusion of choice. But here, we just remove IL-3. So all the cells that don't have an oncogene should die. And the cells that live should, of course, be green because they should have gotten CRE expression. And then these cells we can screen to see whether they indeed have the fusion of choice, but we don't have to screen numerous clones to identify this. And the nice thing is they've been very successful. So with two of the RET constructs that they made, both the TRIM24 and the KIF5B, they were able to isolate BAF3 cells that grew independently without IL-3. And nicely, these cells are exquisitely sensitive to two different RET inhibitors, ponatinib here and cabozantinib here, whereas the parental BAF3 cells in the presence of IL-3 are resistant to these. And so this will allow us to then move to the next step of generating the adenoviruses with these constructs, putting those down the mice of lungs and generating tumors as have been shown for EML4 and ALK. And although I don't have time to go into this because we're running out of time, we plan to use these for a number of different experiments, forward genetic screens to unveil cooperative mutations or signaling pathways using Sleeping Beauty mice where the CRE actually activates the Sleeping Beauty transposase to accelerate tumor genesis, metastasis, et cetera. The same type of screens to look for pathways that regulate tumor intrinsic mediated immune inflammation or avoidance. And then we can also use these to look at the effects of other concomitant genes that we often see in these patients, such as TP53 loss. So we don't really understand. We know that P53 is often a poor prognostic factor for these patients, but we don't understand what effects it has on the cell. And so the CRE in these viruses will allow us to kind of simultaneously knock in an oncogene and knock out a tumor suppressor simultaneously. And so to wrap up, broad conclusions here, but matching oncogene-specific therapies to biomarker-selected patients often results in very improved clinical outcomes for lung and other cancers. We need novel drug platforms to overcome intrinsic resistance with undruggable targets. The study of acquired resistance mechanisms has allowed us to kind of generate new and better single-agent TKIs, but have left us a little bit lacking in terms of combination therapies. And what we're really thinking about now is kind of combination therapies up front to battle this adaptive resistance, eliminate the residual disease, and hopefully get better outcomes for our patients. Again, thanks to my lab and other members that I've highlighted throughout the talk, there are many, many others that I could not fit on the screen here. Clinical colleagues in interventional radiology, surgery, collaborators at the FDA, University of Auckland, and our Polish collaborator, Rafal, our numerous funding sources, including philanthropy from new philanthropy from the Hanlons, who are funding EGFR exon 20 research, and the Burge Endowment that's funding other precision medicine initiatives in the lab. And happy to end there and take any questions that you might have. TJ.
So with the 1,000 compounds, do you think you're getting enough active compounds in the tumor to make kind of the toxic region?
That's a good question. Whether or not there'll be enough drug released kind of in the tumor environment to hit that or not is a question that we'll really only answer in patients. Now, in the mouse models, at least that provided enough proof of principle that we're able to get better responses, right? These are very difficult to drug targets, and I think we won't really know. When you look at lung cancers, the majority are hypoxic. That is an open question whether, areas that are normoxic will actually see that. But my guess is that, normoxic metastases or other areas probably will not have dramatic tumor shrinkage based on the mechanism of this drug. The drug is really inactive until it's cleaved. Bob. Yeah, so the question is in breast cancer for HER2-resistant patients where they're trying CDK4 inhibitors, whether there's any plans in lung cancer. I think it's not been a high priority given the availability of other drugs and drug combinations, but it's a reasonable thing to think about. Yeah. I mean, there is that question. In fact, we're not going to answer this one quite yet, but actually Trevor Bavona, UCSF, and I have a bet. So he wants to. So his data for ALK shows that ALK positive cells and tumors are exquisitely dependent on MEK inhibition. And the question is, do you do an ALK plus a MEK inhibitor in kind of a linear pathway? Or do you take an approach like mine with EGFR? So my argument would be that EGFR probably diversifies signaling. It doesn't only enhance MAP kinase signaling, but also spreads signaling to PI3 kinase and others. In fact, we have a large proteomics project with Eric Hora at Moffitt to kind of look to see what else EGFR is doing just outside of the kind of narrow pathways that we've looked at. And we actually are trying to get a clinical trial where we would randomize to an ALK inhibitor plus an EGFR inhibitor, an ALK inhibitor plus a MEK inhibitor. And so we've got a running bet on which one is better. I think that hitting an upstream target that actually diversifies signaling is probably a better way to go. The problem is once you get down to things like CDK4 inhibition, you're basically talking about chemotherapy, right? You're talking about things that, inhibit rapid cell proliferation. And so you start getting back into the realm of toxicity again because you're non-specifically inhibiting cell proliferation.
The idea is that so many cells don't require
Yeah. Right.
So the idea is that the cancer cells are dependent
Yeah. That's, I mean, something to try. Sorry, can you repeat the question? You know, that's a good question. And I've given a little bit of thought to this. So the problem is once you kill these cells, whether, I guess, are you talking about through so there could be non-specific mechanisms of, necrosis and cell death that might kill neighboring cells. The drug is covalently attached to EGFR molecules, so I'm not sure that once it kind of is activated in a cell and binds an EGFR molecule, whether it would be freely available even if that cell was killed and the contents were released, whether that drug would be active against other cells around it. We just don't know. But I mean, again, the concept that it is activated in hypoxic regions, I think, is proven by the fact that it's actually a pretty modest in vitro inhibitor under normoxic conditions. But as soon as you put it in a living model, the drug gets activated and shrinks tumors. Now, how much it does that and for how long is an open question. Yeah, possibly.
Our standard protocol for the resistance patients is to do 2 or 3 biopsies on follow-up. And what we do with those is mainly next-gen sequencing to find the resistance mechanism.
Yeah.
Is that adequate? What's our yield?
So we've talked about different approaches. So again, going back, I have colleagues at UCSF who are going to be doing whole exome sequencing on their early rebiopsies. I think that would give you some interesting data on maybe the emergence of early clones, but I think that the clones are so low frequency that we're not going to necessarily see much clonal evolution at 2 or even 4 weeks. That there's just not much going on there. So our preferred approach is going to be twofold. One is RNA-seq as the primary kind of global approach to look at transcriptional reprogramming, and that's based on some of the work that Lynn's done in early treatment of in vitro cells and tumor models with EGFR mutant cancers. But then we're also going to do some focus work, so using the proximity ligation assays, for example, to look at proteomic signaling that occurs early on and changes that occur early on. So RNA-seq is going to be the primary one. I don't think that kind of next generation sequencing is going to yield much. We know some of the we get the clonal evolution at acquired resistance. We're not really looking for those answers. We kind of know a lot of those already. What I'm looking for are the vulnerabilities early on that aren't detectable by, sequencing of a mutation. All right.